Principles of Setting Up a Molecular Organizer, Primary versus Secondary by Caroline Zhang. When an embryonic zygote develops, every cell within it has identical genomes, yet they are able to diversify into many different fates due to gradients of mRNA and proteins. Furthermore, cells are able to differentiate even more complexly as these gradients send certain cells along a particular path of gene expression, setting the stage for those cells to begin influencing nearby cells. In other words, cell intrinsic signals lay the foundation for cell cell interactions, using signaling molecules to further guide nearby cells of the embryo. An organizer is formally defined as a group of cells in an embryo that can induce and pattern adjacent embryonic cells. Let's first take a look at primary organizers. A node is an enlarged group of cells located in the anterior portion of the primitive streak in a developing gastrula, where gastrulation first begins. The node determines and patterns the anterior posterior axis of the embryo by directing the development of the chorda mesoderm, a specific type of mesoderm that will differentiate into the notochord, somites, and neural tube. Victor Henson first noticed Henson's node in chick embryos in 1875. However, German embryologist and Nobel laureate Hans Spemann and his PhD student Hilde Mangold were the first to introduce the term organizer when they published their work on the node in Xenopus frogs. In 1924, Mangold performed an experiment where she transplanted a piece of tissue from the gray crescent region of one newt gastrula into the ventral side of a second newt gastrula. The transplanted tissue developed into a second notochord and neural folds, going on to form a second central nervous system and eventually a two-headed tadpole. Most remarkably, the neural folds were built from recipient cells, not donor cells, meaning the transplant had altered the fate of the overlying cells so that they produced a second head. Spemann and Mangold used the term induction for the ability of one group of cells to influence the fate of another, and called the dorsal lip region the organizer. Henson's node was soon found to have nearly the same characteristics. In the 1930s, Conrad Hall Waddington observed the formation of notochords, somites, and neural tube when Henson's node was transplanted into another area of the chick, suggesting that Henson's node too served as an organizer. But how do these organizers work? In Spearman's organizer, expression of organizer genes depends first on Wnt transcription factors whose mRNAs were deposited by the mother in the vegetal pole of the egg, which migrate into the gray crescent region where they are translated into Wnt protein. Dorsal Wnt accumulation induces the activity of nodal, a member of the TGF beta family, which induces expression of the proteins of Spearman's organizer. Many studies have searched for the organizer proteins that induce nerve tissue, but only recently has it become clear that the organizer does not induce the central nervous system, but instead inhibits epidermis inducing signals originating from the ventral side of the blastula. Ventral cells secrete a variety of proteins such as BMP4, which induce the ectoderm above to become epidermis. The Spearman organizer blocks BMP4 action by secreting chordin and noggin, which bind to BMP4 and prevent it from binding to receptors on the overlying ectoderm cells. This allows the cells to follow their intrinsic path and form neural folds and eventually the brain and spinal cord. Henson's node functions similarly. VEG1 and nodal, expressed in the posterior part of Collar's sickle, work together to pattern the AP axis and form Henson's node. In chicks, Henson's node secretes chordin, noggin, and nodal, which dorsalize the mesoderm to induce neuroectoderm, as well as FGFs, also critical for signaling. However, much is still unknown about the mechanisms of Henson's node, and it continues to be a source for new information regarding axis specification and notochord development. While the global mechanisms of Wnt and BMP gradients can establish a crude initial patterning along the AP and medial-lateral axes, this patterning is increasingly refined through secondary organisms in the neuroepithelium at the neural plate and neural tube stages. The DV axis is patterned by two major signaling centers at opposite poles of the neural tube. The dorsal roof plate, a specialized cell population along the dorsal midline which secretes BMPs and Wnts to pattern the dorsal neural tube, and the ventral floor plate, a strip of wedge-shaped glial cells along the central midline of the neural tube, which secretes sonic hedgehog to induce ventral cell identities. Both are induced by molecular signals from the epidermis and notochord respectively. The notochord itself is an organizer that can mediate ventral neural patterning. In contrast, the AP axis is patterned by several discrete local organizers, including the mid-hindbrain boundary, anterior neural boundary, zona limitans intrathalamica, and rhombomer boundaries. These express signaling factors such as Wnt, FGF, Wnt inhibitors, and SHH, and have been shown to have organizer function through different transplantation and knockdown experiments. As discussed before, secondary organizers influence cell fate in surrounding tissues by secreting diffusible signaling factors that often act in a morphogen like fashion, inducing different responses and receiving cells at different distances from the source. In addition to this defining feature of organizers, several other commonalities have been observed. 1. Organizers form along cell lineage restriction boundaries. Boundaries have been discovered to often coincide with organizers, likely due to two reasons. They minimize contact between flanking cell populations, which may help to keep organizers in a straight line, facilitating the generation of a consistent diffusion gradient. And cells on either side are kept in separate immiscible pools, thereby stabilizing a pattern after it has been induced. 2. Positive feedback maintains organizers. Another feature shared by several neuroepithelial organizers is that their maintenance depends on the signal they produce. Both the floor plate and the roof plate are induced by their own signals, BMP and SHH, and the ZLI depends on ongoing SHH signaling. 3. Intrinsic factors regulate differential responses to organizer signals. How can one signal induce asymmetric responses on either side of an organizer? Ectopic expression of competence factors IRX2 and IRX3 are dependent on the organizer signals FGF and SHH and convey a pre-pattern that determines the competence of different subdivisions of the neural tube to respond to secreted signals. 4. HES genes prevent neurogenesis in organizer regions. Organizers typically coincide with boundaries characterized by slower proliferation and absence of neurogenesis. In boundary regions, strong persistent expression of HES genes, inhibitors of neurogenesis, sets boundaries apart and allows organizer regions to form. So, what are the key differences between primary and secondary organizers? As mentioned before, the primary organizer is a global mechanism that acts early on in development, whereas secondary organizers are local organizers that arise later on to refine differentiation. In addition, primary neural induction is largely vertical, since it involves signals coming up from a different embryonic level, i.e., axiomesoderm or endoderm, which act upon the ectoderm, while secondary organizers develop as specializations of the early neuroepithelium, and their signals act mainly by diffusion or propagation by cell-cell contact in the plane of the neural wall, i.e., planar effects. Thank you.